This is episode 273 of the Stem Cell Podcast, Hematopoiesis and Cancer, with the legend, Dr. Irving Weissman. Hey, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we're talking to Dr. Weissman from Stanford University. He's on the podcast to talk about his illustrious career, studying a lot of things, amongst them hematopoiesis and cancer. We've also got our usual roundup of recent highlights in stem cell news. That's coming right up.
But first, and on the topic of Irv, we'd like to remind our listeners about Hematopoiesis News, one of Stem Cell's free weekly scientific newsletters. Save time and keep current with Hematopoiesis News. You can subscribe for free at hematopoiesisnews.com.
All right. Well, by the time this hits the air, it will be a few weeks out from the ISSCR 2024. And we had a really fantastic time. I mean, recorded some delegate stuff. We did the daily roundup. We had that live recording, which was outstanding. Generally a fantastic experience across the board. And you know, the science, not to mention the science, which was super rich, and the site, Hamburg, which was an excellent host for the meeting. I'm starting off the roundup with a little bit of, I guess, residue from that meeting in terms of the science. I'm talking about somites, which was, you know, we had a side session last meeting, I think it was, maybe even 2 ago. I think it was last meeting from Porquier Lab about these somitoids, segmentoids from Meow et al., which was eventually published in Nature. We had Olivier Porquier on the show. We talked about it. This is a bit of a callback to that idea in terms of the somite. You know, the formation of somites, that's what dictates the body plan, right? In an embryo, and ultimately structures the musculoskeletal system. There've been a lot of theories from embryo knockout models and everything, all types of species, to try and fill in dynamics of somatogenesis in terms of the signaling landscape, showing that there's gradients of, you know, FGF, Wnt, retinoic acid signaling along the rostrocaudal axis in the presemitic mesoderm. But the mechanisms of how the boundaries are established remain a bit elusive, mostly because the models are inaccessible, right, in terms of real time, and particularly how you can modulate the morphogen gradients. You know, that's the key thing lacking in any of these knockout models, is how do you recapitulate those gradients, particularly in mammalian species. You know, they're in all vertebrates, you get the somatogenesis, and you can study it in these, you know, in fish, for example. But in mammals, which we care about knowing, it's pretty inaccessible. And in particular, it's the segmentation aspect and the boundary establishment and the mechanical forces that are driving that and are responsible for the delamination of the nascent somites from that presemitic mesoderm following lineage fate specification. That's not really well understood. And it's important if we want to understand what regulates somite size in across all mammalian species, but particularly in ourselves, and understanding stem cell differentiation systems, etc., as we move closer to these organoid models that recapitulate a lot of the anatomy of organogenesis. And then there was the Porquier Lab, Meow et al. This was a real bombshell showing that you could recapitulate the somatogenesis in these HPSC-based models. But even so, those were free floating, right? That showed somite formation, but they didn't control, again, those morphogen gradients or the biomechanics, you know, the strictures of the matrix that have a large part to do with how these somites are formed. So step in uh, Jianping Fu, who actually I caught his session. If you followed the day by day roundups on day 4, I reported on the story from John Ping Fu's lab about the neural tube model, where they had these beautiful chip channels where they recapitulated the morphine degraders. He didn't talk about this at all, even though it was published. I think July 8th, so right before the meeting. He didn't have a session on this at all, but he essentially applied that same system to this Porquier. Porquier is an author here, as well as Yun Chuan Miao, the first author on that Nature paper a year ago. But it's incorporating that somatogenesis segmentoid uh, model into the same kind of channels that he described in the neural tube model. Um, and effectively, yeah, what they did is they confined the pre-Semitic mesoderm that they generated from HPSCs into these trenches, these microfab trenches that were the same base for the neural tube, and then they overlay this microfluidic morphogen gradient on that, and cause this axial patterning, and there was spontaneous formation of the rostral to caudal somite formation. And it's beautiful. I mean, you got to look at this now more than ever with these bioengineering stories. The multimedia data is so critical. The live time lapse imaging here, and then reporters in this paper. It's really a feast for the senses. Um, and here, the key insight and what they were getting at was, yeah, all the morphogen gradients, and showing that they could recapitulate it with known knowledge from a lot of embryo models, in vivo, not like in vitro embryo models, the history of somite research. But they correlated this like biomechanic element, right? And showed the interplay of both like cellular signaling and the tissue biomechanics and cell formation. They had these theories that were able to be, modeled of the size dependency of the somites following from pre-Semitic mesoderm. So a lot of, the first listing here for Dr. Fu is his post in the Department of Mechanical Engineering. Amongst that, also biomedical engineering and cell developmental biology. But first came mechanical engineering, which I think tells you something about his focal point. And really, for me, just underscoring how these microfabrication, microfluidics, recapitulating the scale of developmental biology in embryos, specifically like human embryonic tissues, is leading to, a real explosion in these studies that are creating tissues that have like high fidelity to the anatomy of the physiological correlate. So cool, Arun.
Very cool. And I think we have a nice parallel set of stories here with your first paper and my paper, because we're talking about modeling development or aspects of development using these bioengineered systems. And it's actually not something that's traditionally ascribed to these microfluidic systems. I mean, I have some experience doing some organ on chip work as well. I mean, typically you have a terminally differentiated cell population that you're replating onto these quote chip systems, say model drug toxicity or heart disease or something like that. But here you're taking a look at somatogenesis, those very early aspects of development, like what you're saying, um, using these really beautiful visuals and these fluorescent reporter systems. You got to take a look at some of the videos associated with this particular paper. Videos do they really do it justice here. I think it's a perfect system. It's a really nice system to look at gradients because you can finally assay and evaluate the impact of these morphogen gradients on somatogenesis. As we know, it's a very complicated process in vivo, and you're very significantly simplifying this in this ex vivo system. But the advantages are scalability, the amenability to imaging, and really being able to tease apart those fundamental mechanisms at, like, say, single morphogen, single gradient. You can evaluate this on this chip system. And they allude to it. It's not a perfect system, it's very simplistic. And they want to extend this to other pathways as well. But I think something really nice to build on.
Yeah, I love what you said there. I agree 100%. And Um, yeah, getting back to the capability, both like how far it's advanced, but also how far it has to go. I mean, you think of the history of these organ on chip models, and now even 5 years ago seems really rudimentary. And probably similarly, looking back on this, the idea will be like, yeah, well, you're not able to really model the most sophisticated, like intracellular signaling modules that are happening across the many dimensions of the tissue. But on the other side of that, I think what we're learning with these bioengineering systems, just from a practical standpoint, is like how robust. We already knew this. Embryogenesis is so robust, and given the right environment, given the right foundation. And here it's this idea of you can just get the ball rolling with a relatively simple overlay of these known gradients, and then the cells, you know, for the putting it simply, take care of it from there. And yeah, there's still a long way to go in increasing the complexity of these. But I think both from a practical standpoint of how we can create better tissues, and also from the standpoint of like we could probably integrate a few more dimensions and temporal, spatial temporal modulations here to improve these systems even more. So I'm super excited about the next phase here. This is essentially one modus here, the model, neural tube and somatogenesis, showing how these base technologies can be applied across different fields of study. But I think there's still a lot more sophistication that FULAB and others are going to bring to bear.
Absolutely. And to perhaps go on a bit of a tangent or a soapbox, whatever you want to call it, there's folks out there who think that, the organ on chip system is powerful enough that it, and perhaps advanced enough that it can replace in vivo models entirely. I am far from that group of people. I think there's a whole bunch of avenues and areas where in vivo models are extremely useful. When you're talking about drug toxicity and looking at the effect of a single drug on potentially a single cardiomyocyte or single cell type like a cardiomyocyte, then perhaps the organ on chip system can be used in the place of or to supplement the in vivo cultures. But if you're talking about something as dynamic and as complex as in vivo, as development, um, I don't think you can replace that in vivo system. I think this is supplementing what you would have with, say, a mouse model or something that would better approximate that in vivo developmental processes that are undergoing in humans. And like we're talking about, this is a uni or bidimensional system. And there's a lot of variables that can only be, in my mind, only be modeled by an in vivo system. So in the context of development, I don't think these chips are going to replace mouse models or animal models anytime soon. So moving on to another study that's kind of similar. We're talking about these microfluidic systems to model development and processes of development. Again, not something that they're traditionally utilized for. Typically, they're used for drug toxicity screening, adult phenotypes, adult diseases. But once again, here, they're modeling a developmental process in the development of the intestine. Okay. And they're looking at the role of interstitial flow, this really important concept in intestinal function. They're trying to model that impact of interstitial flow on the development of the intestine. Okay. So there have been a lot, and I mean a lot, of gut on chip approaches that have been published over the last 5 years. And in fact, there are folks here at Cedar Sinai, where I work, who have published on this topic extensively. We have our own microfluidic systems in house. And typically, what we do is we make organoids, gut organoids, IPS derived intestinal organoids, and then place those on the chip. And some of the cool things that we've seen is that by adding active fluid flow, you can really dramatically enhance the proliferation and the maturation of these stem cell derived gut cells or primary gut cells on the chip system. But one thing we haven't done, and I don't think a lot of people have done, is again looking at how these things develop on a chip. So that's actually what they're doing here, this is the lab of Kazu Takayama over at Sira, the kingdom of Shinya Yamanaka. Just kidding. Over there in Japan at Kyoto University. Shinya and Friends, we can call it that. This is a cell stem cell paper titled Construction of Multi-Layered Small Intestine-like Tissue by Reproducing in Interstitial Flow. Okay, so here they're trying to recapitulate that process of intestinal development, and they suspected that interstitial flow within the intestine, which is powered by the circulating blood plasma during organogenesis, is a vital factor during intestinal development, and in particular to be actually accurately produce the structural and functional characteristics of that intestine. So here they're constructing this multi-layered small intestinal tissue by incorporating into interstitial flow into this organ on chip system, um, by differentiating the cells, the iPSCs, into endoderm and then intestinal cells on the chip itself in the presence or absence of the interstitial flow. Okay. And then they showed that indeed, by differentiating the cells, approximating development on a chip, um, with the interstitial flow, it was able to further enhance the maturation of the cells, lead to the development of this really nice three-dimensional small intestine-like tissue with some beautiful villi-like epithelial cells and structures, which, they did some great electron microscopy to really confirm and compare these villi-like epithelia with the real deal. Also, they had a aligned mesenchymal layer. So ultimately, it's a cool developmental analog for the development of the intestine, which can perhaps overcome some of the limitations of conventional intestinal models. Again, you have this introduction of the biomechanics. That's one thing that the traditional intestinal organoids don't have. They don't have the active fluid flow. And as we know, for various cell types, you need that biomechanical shear stress to further enhance the maturation of these cells. This is not just intestine. This is the case for, as you know, endothelial cells, for vasculature. These things are very responsive to mechanical shear stress. And, I think they're demonstrating here that it's critical for the maturation of these cells over the course of intestinal development.
I mean, talking about this, I'm with you that we're not going to replace in vivo models. By the way, I just want to throw that out there. At least we cannot rely solely on in vitro models, and I think this is a great illustration of why that is. The story I talked about from the Foo Lab kind of set up how the structure and order of the system can create these mechanical forces that are essential to development and dictating form. And here it's the correlate, right? Is that the force itself is dictating the structure of the tissue and the differentiation and specialization, maturation, all those things, activation. So yeah, force. We talk about force all the time, like it's a thing, but here I think it really underscores how in vitro models can only hope to can only aspire towards to approximate those forces. When I'm looking at Matthias Lutov, who swept the ISSCR a little bit, with his in vitro generated structured gut models, building them out as opposed to like growing them in, but a combination of both. You see how force is really critical just to like get rid of all the dead cells, right? Flow just for dynamic exchange there. In this case, for a lot of other things, and the intersection of force and fate in stem cell biology is paramount. And I think that while we can model a lot of these things, it really just shows how amazingly complex embryogenesis is. While robust, there's so many forces and influences that come into play. It makes it seem like we're never going to be able to get the ectogenesis thing going.
Yeah, and then you do something crazy like take gravity out of the equation, right? Like we were talking about at the ISSCR, and there's a mechanical force that is influencing all of our lives, but we take it for granted. And things go out the window when you take it away, right? So development and how you can manipulate organoids, all these kind of things can be altered in a microgravity context, something we talked about with, Kat Jameson, Davide Maroto on our live show. But you're right. The other thing that actually Clive Svensson, my boss, talked about at ISSCR was that, when we're traditionally doing our cell cultures and the ways been doing them forever, it's almost like we're growing our cells in a static format in a pond, like what he's alluding to, just like in a pond of cells. And as we know, in reality, it's not a pond. You have very active biomechanical stress going everywhere in your body, whether it's interstitial flow in the intestine, whether you have circulatory shear stress. Cells are dynamic, and cells respond to dynamic shear stresses and biomechanical stresses in unique ways, right?
Yeah. All these models are wrong. They're dead wrong. Not dead wrong, a bit wrong. Still useful though. We talk a lot about the Holy Grail on the show, the Holy Grail of blood, the Holy Grail, everything, right? But like quite literally, the Holy Grail in like the Indiana Jones sense is a kind of fountain of youth, right? Or a rejuvenative factor. Since the days of, was that Ponce de Leon, Fountain of Youth? I don't know. Fountain of Youth. It's like an old story. And it's been, I think, relegated to the realm of fantasy, pseudoscience, maybe even. But over the last few decades, we've taken the pseudo off of there, and the longevity research, as it's called, is really picking up a lot of momentum. And it's a deep well. There's a ton of signaling pathways and mechanisms of modulating lifespan across species. Observationally, ERK signaling, mTOR, IGF, insulin has all been implicated in aging and dictating the hallmarks of aging, including mitochondrial dysfunction, inflammation, cellular senescence. And then there's like the whole therapeutic element of that, talking about caloric restriction, sirtuins, red wine, resveratrol. I think I said it right. And more recently, a lot of mouse models, the GDF11. It's there's a ton of studies out there, specifically in like yeast, worms, flies, extensive research. It's tough with the primates because they're long lived already. So those studies are ongoing for probably decades. Um, but one key facet of those studies is like, yeah, is increased lifespan. Is it increased health span? I think that's the question that we all are getting at. I don't know if you've seen any of these caloric restriction people, but they look rough. Which is not to say they're not healthy. They don't look happy, if I'm being honest. Mice are a great model as opposed to like yeast, worms, fruit flies, because they're the aging pathologies that are like clinically relevant and observable. They manifest in mice, right? So mice are a great model. And in recent years, inflammation and its relationship to aging pathologies has been increasingly recognized and prominent. And it's not just that like inflammation is a hallmark of aging. Like, oh, you see increased inflammation in old age. It's now increasingly becoming clear that inflammation is driving some of those pathologies, as you might expect. And that's due to like dysfunctional adaptive immune system, where there's like immunosenescence, as they call it, but also inappropriate activation of like innate inflammatory immunity, such as like an IL-6 signaling. And there have been some therapeutic inroads. NF-kappa B, JAK-STAT signaling has been implicated in aging, and these JAK inhibitors can alleviate that. Here, this is a story from Stuart Cook's lab with Anissa Wijaja, who are at Duke National University of Singapore Medical School. Is that a go Blue Devils? Duke in Singapore? I'll give it to them. Go Blue.
Go Blue
Um,
Devils.
the study here, and I read on the side that this emerged. It was one of those like, oh, look at that things where they saw that IL-11, which is a pro-inflammatory, pro-fibrotic member of the IL-6 signaling family, they found that in aged mice, like IL-1 was really cooking. That was, I think, the primary observation. I think I'm getting that right. That drove this study, and led them to look into and really dissect the relationship of IL-11 and signaling downstream of it with aging. And what they found is that indeed IL-1 upregulated across tissues. That it regulates ERK, AMP kinase, mTOR access at the cellular tissue, at the organism level. It modulates these aging pathologies. They showed that if you delete IL-11 or the receptor, you can protect against metabolic decline, frailty in old age. This is crazy here, which is why people are going to be clamoring for some anti-IL-11. But they show that when you administer the anti-IL-11 to 75-week-old mice, so those are some geriatric mice, you give it to them for 25 weeks. I'm sure it's very expensive, but it improves metabolism, muscle function, reduces aging biomarkers, and frailty across the sexes. All right, and they do these lifespan studies where they delete and knock out of IL-11. The lives of male and female mice are both extended by an average of 25%. And here again, get me some IL-11. They treat with anti-IL-11 from 75 weeks, all right, until death of these mice, and they find that it extends the median lifespan of males and females by 22 and 25% respectively. So what? I mean, anti-IL-11 therapy, which by the way is already in early clinical trials specifically for fibrotic lung disease, but let's go off label, Arun. I'm not getting any younger, but maybe I am.
Oh man, we got to be careful here. I mean, dramatic. I don't like to over represent results from basic papers, but this is a dramatic result. 25% extension of median lifespan in both male and female mice. Um, wow, that's a long time. I mean, IL-11 is a to simplify these things, right? As you alluded to in the very beginning, we figured out by now that inflammation is bad and blocking it is good. So that's kind of what we're doing here in this paper, right? It's sort of an obvious study. But IL-11 is just one factor in inflammation, and you can look at a billion other factors that perhaps would have the same effect, and maybe they're doing that. So, where does this train end, in terms of blocking inflammation? You talk about the fountain of youth. It goes way back before, what's his name? De Gama? Is that what you said? Ponce de
Ponce
Leon. Ponce de Leon. It goes back to Herodotus in,
Whoa.
the Greek mythology or Greek history. So this is an idea that people have been hunting for a long time now, and I'm not saying we figured out the fountain of youth here, but maybe one drop in the fountain. We could say that.
Wow, what a I mean, first, beautiful metaphor. I have to give you that, and thanks for the little history lesson.
You're
Um,
welcome.
I'll say you're right, though. You're right. What you're getting at is so right, which is that we've been looking for this thing forever, and you often find what you're looking for in science. So this is a drop in the well, I'll say, but I'm encouraged just by the idea, one, that although there's all these things that converge on aging and inflammation, and it's a simple idea. Here's like one thing in a not saying it's a silver bullet, but here's a single point therapy that seems to be having an effect in mice. And of course, I think that's the big question, right? Is when you take these people, I mean, are these people with fibrotic lung disease winning, Arun? I mean, I shouldn't even joke because I would not beg for fibrotic disease of any sort in order to justify IL-11 therapy. But I think the point I'm trying to make there is like, I wonder if there's I'm sure they're drawing up the clinical trial right now standalone, but I wonder if you could get some early indicators from these patients who are being treated for fibrotic lung disease, maybe looking outside of that compartment and seeing if there's an influence or rejuvenative effect in perhaps the blood. I don't know. But I'm encouraged because although it's mice, 75 weeks is like a dead mouse, pretty much, as far as I'm concerned. For them to go on past that, to just start the therapy, then I think there's still a chance for me.
Oh man. Yeah, 75 weeks. Wow, those are, like you said, geriatric, beyond geriatric mice. People are going to be keeping a very close eye on this. And we've talked a lot about recently on the show about nodes and how single focal points, single pathways, single proteins, and whatever in pathways. If you're able to identify that single node, it's a very powerful thing. But like I said, drop in the fountain. Who knows what other nodes there are out there? And people are going to be staying very closely tied to this. And as somebody who does caloric restriction occasionally and makes, gets really grumpy as a result of it, maybe this is a better alternative. I don't know. We'll see. Moving on to our last paper. This is a resource. It's a Nature paper. It's another one of those single cell atlas papers. And a couple years ago, these were coming out almost every single week, and it felt like we were covering an atlas every single week on the show. It's died down perhaps a bit. Maybe we've atlased the whole body by now. I don't know, but apparently not. This is an important one, though. This is looking at human brain vasculature over the course of development, adulthood, disease, and doing a single cell atlas approach of this. So pretty straightforward. And again, like as you usually get in these atlas approaches, this is a huge group of people. It's led by Ivan Radovanovich over there at Toronto, the Division of Neurosurgery, Department of Surgery, and the University of Toronto. Um, Really an important study here in Nature, uh, doing exactly that, profiling the human brain vasculature, not just in standard development, but in disease as well. We've talked about how vasculature is really important for obviously modeling things like the human blood-brain barrier, but it's critically important just in proper brain function. So what they did here was profiled and single cell RNA sequencing over 600,000 freshly isolated endothelial cells, perivascular cells, other tissue cells, tissue-derived cells from 117 samples, 68 human fetuses, adult patients as well, and created this huge atlas of developing adult control and, importantly, diseased human brain vasculature. I think a lot of the atlases out there, they only focus on the healthy. And I think, at least in the cardiac side of things, I know there have been a lot of dilated cardiomyopathy atlases that have been really helpful in understanding what goes wrong with the heart. And so same idea here. And what they found, in addition to just doing the profiling, doing the seek, I think that's the next step of these atlas papers, right? That I think is really important. If you have the money and resources and time, anybody can do a profile. Like anybody can just do a billion RNA seeks, single cell RNA seeks of endothelial cells or whatever cell type. It's what you find afterwards that I think is really exciting here. And they alluded to that. So they figured out that there's a lot of molecular heterogeneity of the vasculature of healthy fetal and adult human brains across a bunch of different pathologies, 5 vascular dependent central nervous system pathologies, including brain tumors, brain vascular malformations. So it's literally at the endothelial level, the profile of the cells under those different conditions is extremely different, not as homogeneous as you might expect. All right. And there's also specifically alterations of arteriovenous differentiation and also reactivated fetal programming in the disease vasculature. So it's almost like these things are going back in time in a disease state. So these pathological endothelial cells have a loss of central nervous system specific properties. Again, they kind of de-differentiate and upregulate MHC class 2 molecules, which is indicating that these are not typical. These are atypical central nervous system endothelial cells. They also did a bunch of cell-cell interaction analyses, some computational analyses to predict that there's a lot of endothelial to perivascular ligand receptor crosstalk, which is actually important for the proper functioning of the brain itself. And these are in the realms of immune-related angiogenic pathways. This is again, their conclusion here from their analysis is that there's a really central role for the endothelium within proper signaling networks in the brain. So it's not just the neurons, it's also the endothelial cells that are critical for that uh, architectural signaling in the brain. So ultimately, it's more than just a useful resource for the community. I think it's uncovering some fundamental mechanisms as to how the actual brain works in a proper and disease state. And so take a look. These Atlas papers are always publicly available. So if you're working in this area, I'm sure that's going to be super useful.
Very useful, comprehensive reference here, an atlas. And I think the insight is, I wouldn't say profound, but it's consistent with, I think, the revelation long now accepted that endothelium is not just plumbing, right? And also I don't think you could have gleaned that insight, at least in as clean a fashion here, about the reactivation of the fetal programs here, if you hadn't, in a single effort, consolidated the fetal samples, the adult, and as you emphasized, the pathological endothelial brain endothelial samples. So that was it's it. I mean, I think this is probably the reference standard now, and will serve as a essential resource for studies not just of the brain vasculature and perhaps pathology, all numerous pathologies not included in this data set, but also just endothelium generally, and how dynamic it is, and the discrimination of different endothelial subsets, really key. And also, I think a key point here is that I don't know if it was in every sample, but in many of these samples, is that they sorted and enriched for endothelium, but then they also took the unsorted population, because as I just kind of alluded, you find what you're looking for here, and they would have limited the data set to just focus on endothelium. Here, I think that they can identify a bunch of, like, I wouldn't say quasi endothelial per se, but not every endothelial cell is going to have CD31 at the cell surface, right? So I think that it's really critical here that they had a very well thought out approach, evidenced by the authorship here. More than 35 plus authors, including Vivian Tabar. I'm sure these are neurosurgeons across the board, but in order to get them to standardize their methods and to ensure, like, I looked in the methods, they were very careful about, like, the cautery, so that they wouldn't be damaging the cells, and standardizing all the collection and dissociation, etc. I think is really key to making this a clean and highly useful, and will be highly utilized data set.
It's not just plumbing, boss. Like what your guy Shaheen Rafi always says, right?
That's a
It's
very
more than
good
just
impersonation. You did it.
not bad, right? I feel like I've heard it enough on this show where I could do it once, right? Um,
Angiocrine, boss. Angiocrine.
there you go. That's the other one. So like Shaheen says, it's more than just plumbing. There's a lot of mechanistic insights to be gleaned from endothelial cell function, their interaction with other cell types too. The other thing that I think could be interesting here, and I think also in the realm of what Shaheen is interested in, the production of bona fide brain microvascular endothelial cells from IPS, which has been a controversial topic, as you probably are well aware of. And this could be perhaps a nice roadmap in terms of how to accurately recapitulate those elusive cells in a dish. I don't know.
Yes, the elusive BBBEC, but we try and steer clear of controversy on this show. So I'll just wrap it up. Before we get to our really, really fascinating conversation with Dr. Irving Weissman, a living legend, I have a quick message from STEMCELL Technologies, who have been in the field of hematopoietic stem and progenitor research for over 20 years, pretty much when Irv got in the game. And during those 2 decades, STEMCELL Technologies has learned a thing or two. Visit stemcell.com/hemahub for educational resources to help you further your research on hematopoiesis and hematological malignancies. All right, you guys, today we have a very special guest, I think, admired by all you listeners out there, if not all scientists out there in the world, a living legend in stem cell biology, Dr. Irving Weissman, who is the Virginia and Daniel Ludwig Professor of Clinical Cancer Research, also Professor of Pathology and Developmental Biology, and Emeritus Director of the Stanford Institute of Stem Cell Biology and Regenerative Medicine at Stanford University. As the first scientist to identify and isolate mammalian blood-forming stem cells in mice, Dr. Weissman is considered the father of hematopoiesis. His work has contributed to the understanding of how a single hematopoietic stem cell can give rise to specialized blood cells throughout our lifespan. Dr. Weissman is also a leading expert in the field of cancer stem cell biology, where his work sheds light on the understanding and pathogenesis of multiple human malignancies. Moreover, he's led the development of some of the most pioneering approaches for cancer treatment, including anti-CD47 antibodies that can eliminate cancer cells in mouse models. And that's just to name a few of his innovations. There's really a long list there, which maybe we'll come back around to, but I doubt it, because there's just too many to talk about within the scope of this interview. But we'll get some good kernels from Herb, I'm hoping and I'm assured. Dr. Weissman, Herb, thank you so much for joining us on the show today.
Thank you.
Yeah, thanks for being here, Irv. It's always a pleasure. I mean, it seems like you've done it all, but at the same time, there's still so much for you still to do, especially in the world of hematopoietic stem cell biology. I mean, we could talk forever about what you've already done, but let's stay in the present and the future. So for the handful of people in the stem cell field who have been living under a rock and don't know about you and your lab, so could you talk about like what you're working on now and at Stanford and maybe the areas of HSC biology that you're the most excited about?
Sure. So, as you said, the blood-forming stem cell, hematopoietic stem cell, sometimes we call it HSC, is the cell, the only cell in blood formation that self-renews. So all of the daughter cells from the stem cell divide a certain number of times while they differentiate. So you can give, and we did this once a long time ago, you can give back to an irradiated mouse all of the cells in bone marrow except the stem cells, and they'll die. If you give them just the stem cells, which are 1 in 100,000 cells of the bone marrow, so long as you give 50 to 100 of them, they'll survive. And they'll survive because they make the donor-derived cells. Now, I would say the things that we're working on now that are exciting have to do not only with the science, but also the translation of the science. And I think this field is a good instruction of how you can, and I believe should, think about translation when you make a discovery. I think in general, I would say the time that you make a discovery that starts to open a field, you're the only person on earth who has at least some part of the understanding to know what the possibilities would be for therapeutics or if you wanted to commercialize it. And also, probably the only one who could ferret out what happens when things go wrong. And plenty of times, and especially in clinical trials, they hit the things that go wrong before they hit the things that go right, which is an interesting topic. So I would say right now, a very early finding that in fact got me into stem cells was the induction of immunological tolerance by Peter Medawar and Rupert Billingham and Leslie Brent, 1953. In 1956, I was a high school student in Great Falls, Montana, and had begun working in the lab of a transplantation immunologist / pathologist. He did so much pathology that I almost got to do my own experiments from the beginning in high school. And I'll just say as an aside, because it is one of my later points, I never got into the top 10% of any class from grade school, intermediate school, high school, college, medical school. And I think that's largely as I lacked the self-discipline, to study so hard you could think of the future getting in. So I just found out in that setting and reading the paper by Meadow et al. That you don't need any of that memorization that you do with all that self-discipline to become a scientist. In fact, it's counter to becoming a scientist because you actually believe what you read in textbooks. And that's the worst thing to do as a scientist. You need to assume that everything's wrong and that what you're seeing is real and you got to observe it. So that's part of that diatribe. But when I was in Great Falls, Montana, even though it was a remote place, Ray Owen and Leslie Brent came through. And as I was doing work on tolerance, I got to talk to both of them. Ray Owen actually started the whole field of hematology and the most important part of transplantation, immunology. He observed as a student in Wisconsin in 1945 that natural parabiont cattle, ones that share a placenta, so-called free martin cattle, were chimeric with their own blood, siblings' blood for life. If they had been born by different mothers or didn't share a placenta, they would be immunized by a transfusion of red cells from one into the other. But he showed they weren't. They kept them. And so he knew enough to say in 1945, there must be something important that keeps making blood after birth. And second, there must be something important about the immune system, that everything is self until sometime after birth. And then it becomes non-self, anything new that comes in. And I thought to myself, well, the development of the blood forming and the immune system is going to be really neat to study. And one way or another, that's what I study, and that enabled us to find the stem cell and so on. So one of the experiments that I did 30 years ago was to transplant mouse blood-forming stem cells from C57 black to BAL, complete MHC mismatch. Of course, the recipients were irradiated. You got them next to death. But I found if they became chimeric, that day that you transplant, or 6 months later, you could transplant a heart, skin, insulin-producing islets from the donor strain, but not a third-party strain, and you need no immunosuppression. So stem cells are not only important for blood formation, and when things go wrong, things like leukemia or immune deficiency or autoimmunity, but they're key for the induction and maintenance of immunological tolerance. So all of my scientific life, I've been thinking, well, we got stem cells for the blood forming system. That about 1988, 86 to 88, we did that. And if we had other tissue stem cells from the same donor, instead of waiting for somebody to get shot in the head, so you get their liver and heart and lungs, skin, islets, you might be able to do it with stem cells. And of course, when embryonic stem cells, or pluripotent stem cells came along, that opened the door that you might have, instead of a person with a dead brain, a cell line that makes hematopoietic and, say, brain stem cells. In fact, the second stem cell that we isolated, Nobuko Uchida, my wife Ann Sakamoto, Rusty Gage, and I, was the human fetal brain stem cell. We published that in 2000. We did it at a commercial outfit. And so we weren't permitted to do the science that we wanted to do to fill in all of the properties of the human brain stem cells. So when the company moved on, Dan Liu, MD, PhD student in my lab, and I went at it again, and now we could do it completely. We published a paper in Cell a year ago where we found that we could isolate using exactly the same system that we used for hematopoietic stem cell, essentially having a cell sorter, a bunch of antibodies, a source of the cells, and then you do what you always did, playing 20 questions, ask a question that divides the population in half, test them by transplant, and then move on to the one that worked and get an antibody that divides them in half. And as you know, 20 questions, 2 to the 20th, it means you get anything that's 1 in a million. And hematopoietic and neural stem cells are much less than 1 in a million. So I know it sounds like we invented some fantastic physical method, with an understanding of electronics or something else, but no, it was just 20 questions.
Mm. Wow. I mean, that's I don't want to say you're oversimplifying, or because you always provide great depth and synthesis, but I think this wasn't the 20 questions I used to play on the long car trips. I'll tell you that much at least. But I think, I mean, the best thing to me about that answer, there's so many parts of it, but one is that, yes, sometimes you got to put something not on the shelf, but you come back around, right? And over the course of what, 60 plus years, you've done so much. And the other part of that answer, which I love, is your statement there that you gotta ignore everything you read in the textbooks, right? And reconcile what's right in front of you, right? The observation that you're looking at, that's the only true thing that you're sure of. And that's, I mean, over the course of decades, there have been so many of those observations that you've made. And the impact of you reconciling them has been monumental, not just in the scale, the number of discoveries, but in the scope. You're talking about fetal brain cells, godfather of hematopoiesis. You're all over the place. You follow your interests and your passion. You've also led this fascinating life, right? And had an outsized impact on a lot of other lives, both within and outside of science and medicine. So with all of that experience and getting back to that idea, the only true thing is what's right in front of you. What's been the most surprising observation or reconciliation thereof that you've had, or alternatively, what's the most surprising thing about the progress of our field, stem cells, stem cell biology?
I'll pick the latter one because, and I'll maybe come back to the former one. So when we had isolated blood-forming stem cells in mice in 1988, I'd had some experience in advising a couple of biotechs. And I knew that the only people who are going to transplant hematopoietic stem cells are people who are in the industry, bone marrow transplant. So we started a company, we had a total of $10 million that wouldn't take you anywhere now. But we actually got to the human blood-forming stem cell when Mike McCune, then a postdoc with me, said, we're not going to be able to find out the function of blood-forming stem cells, even if we isolate them, if we get them from humans, because it's not a whole bunch of medical students who will offer to be irradiated so we could test our cells, excuse me. So we went on to, he said, why don't we put the blood-forming system of a human into a mouse? I said, gee, we tried that a long time ago, and I couldn't get any Till-McCulloch spleen colonies, so I quit. But he persisted, and we went through everything that you had to do not to induce women to have an abortion so that we can use the fetal remains for medical research. And I'm putting it that way pretty carefully because nowadays it's even more serious, the political and personal consequences of saying something like that. But we did it. We made what's called SCID-HU, severe combined immune deficient, humanized mice. And we put in human fetal bone, human fetal liver, under the kidney capsule with the fetal liver, human fetal thymus and spleen. And we could do the whole circuit. We give them a low dose radiation, transplanted our candidate human blood-forming stem cells. And within a year to a year and a half of actually doing those things, we not only showed what was the phenotype, of human blood-forming stem cells that could get out of any mobilized blood or bone marrow, but that they could transplant. And if there were any T cells left in the bone marrow or the mobilized blood, if you didn't get rid of all of the T cells, the skid-Hugh mouse would get graft versus host disease from those T cells. And you know, what happens over and over and over again, it's just one small side trip. I had a grant rejected by the agency that I helped invent, California Institute of Regenerative Medicine. I wrote Prop 71, the science and clinical part. Bob Klein wrote the political part. And it was repeated in Prop 14 in 2020. And they give out grants that, as we said, should allow academics to take things forward all the way to clinical. Well, our isolation of human hematopoietic stem cells back in 1992, the company was Systemics. That company was bought by a large pharma. They needed to have cell sorters. They wanted to do it for various reasons. They never did a thing. Mm-hmm. They didn't call us up. We were gone. They bought the whole company. So I realized for the first time that it wasn't the function of a company to advance medicine. It was like my dad told me, a junk dealer and fur trader in Montana. The function of a company, the only function of a company is to make a profit. And that's a lesson I've learned over and over again. I used to hate it that companies would do it, but it's better to understand that, because then you can try to do something to get around it. Well, CERM, Prop 71 was my way to get around it, because we allowed the state agency to fund research at an academic setting, and then to fund, if they wanted to make the organization all the way to phase 2 clinical trials without having to commercialize. So when that large pharma stopped the clinical trials, we had already done one very interesting trial. We had isolated from the mobilized blood of women who have metastatic breast cancer. And by the way, it usually metastasizes beyond the lymph nodes via vascular seeding of the bone marrow and the bone. So we did it with people who had authentic metastasis, and for the most part, bone and bone marrow were included in the metastasis. So when you look at their mobilized blood, it has plenty of cancer cells in that mobilized blood. And that whole thing that was the craze and then the huge collapse of the 1990s was that the bone marrow transplanters didn't read our paper that eventually came out. They only transplanted mobilized blood, they said, because we know mobilized blood are stem cells. And then when 34 selection came along, they said 34 are stem cells. You'll see it everywhere. You'll see every journal still publishes stem cell in the title, but we're the only ones who ever transplanted stem cells, hematopoietic stem cells in humans. So that's a big lesson. All of those people who memorized their way to leading clinical positions now became our second major barrier. One was commercialization. I didn't understand it. The second was the big barrier are the people who think they understand, who are adamant they understand, but they don't understand. All of what they learned is fantastic for medicine. They memorized all the stuff you're supposed to. So you see that, you do that. It's not even, you don't have to think, it happens. And so that I'm grateful that that developed, but they're not going to be the people who know what to do next. because they don't know anything about how it works in the body. So that's my diatribe part of it. But when I put the grant in 2023 to CERN, I got the 64th lowest score out of 65. And they said, this is going to be a waste of time and money because everybody knows CD34 cells are stem cells and they'll work in metastatic breast cancer. And this is even despite we published the follow-up of that clinical trial. So we had 1996 to 1998, we did the isolation, high-dose chemotherapy, so then you could rescue them with their own blood-forming stem cells. And we showed they were 250,000-fold depleted of breast cancer cells, and also T cells, by the way. All we had in the tube, that little 100 microliters that looked like clear saline, were enough stem cells to bring back white cells by 10 days, platelets by 13 days. Okay? And we were the first people who ever rescued people from that very high dose chemotherapy with cancer-free hematopoietic stem cells. We changed the median survival of that small pilot study from the standard of care 2 years to 10 years. That's pretty good. And by 15 years, and now 26, 27 years later, 1/3 are alive without disease. I tell that to the breast cancer doctors, and they say, no, we're doing better. We've got this. I said, what's your survival at 10 years? Well, of course, what we've done is we've taken women to a less painful death. They don't have to go through high-dose chemotherapy, but they all die. And I got 1/3 alive who had whole lives with their kids, their work, their life. And it doesn't convince them because they've been told by the experts, you do palliative intent therapy only. So at least, I'm not quiet. I told CERN they were full of it and that they ought to read the papers. And then I got funded for at least part of the development. So I've spent a greater part of the last, not a greater part, a significant amount of time in the last 10 years to get that company to let me use the hybridomas we made to isolate stem cells. And they couldn't make a profit on it. And they agreed. Then I got Stanford, almost a commercial organization, don't say that, to agree to be the setting where there would be no extra cost for the antibody separation of stem cells. There still would be cost around other stuff, but they would have a not-for-profit setting. So we got that. We got the hybridomas. We've remade them. We've spent a lot of our own personal money just because I don't want anybody to get rights. I want it to stay not-for-profit. So it happens. And so we are gearing up over the next couple of years to do breast cancer again. And then because there's no T cells, to start in on SCID, maybe lupus, any of the various genetic diseases of the blood-forming and immune system. Sickle cell would be perfect. Now, every time somebody gets a tiny percentage of engraftment with gene editing, it's headlines every time. An allotransplant that doesn't have GVH, that generates even better, we can't get them to answer the phone. It's an important lesson to young people in science who want to translate their discoveries, is that you can't be cowed by those people. They're wrong. And you just have to keep saying they're wrong. And you have to keep going at it and find ways to get there. So we're going to get there, but it's too bad it's delayed. because it means that people died needlessly
Mm.
of a disease they didn't need to die of. So that's the preaching part. The second part that is very exciting is that we all accepted that you give high-dose chemo or you give radiation to prepare a recipient to have space for a stem cell transplant. You got to have also, by that radiation or chemo, you got to get rid of T cells and NK cells that would reject the stem cells when they came in. Well, we started that with Anish Kachekovich and I in 2007 with an anti-C kit to deplete in skid mice these stem cells and found we could transplant them. But when we found out you try to do that even with autologous or syngeneic transplant, if you try to do that with the antibodies alone, it didn't work in immune competent. So to make a long story short, we have many papers over the years. I think the best one, the final one, was Benson, George, and I in Cell Stem Cell 2019, where any donor mouse, any recipient, we just give a cocktail of antibodies. 9 days later, we inject stem cells, same number that we would inject, or maybe some more than we normally would inject. And we get chimerism, and they're permanently immunologically tolerant to heart grafts from the stem cell donor strain, but not others. And it works in full allo, but it also works in haplo. So that means you don't have to have 6 to 8 kids to have a chance of having a match amongst the kids, 25% HLA match. Everybody has either a parent or a child or a sibling who can be a blood-forming stem cell donor. So these are getting over real barriers to try to get there. And the science as you go along tells you a lot. So the most recent discovery that you mentioned is that we found, and many labs find, and by the way, the late Connie Eves was one of those early people who found it. And the late Krista Muller-Seberg, who'd been my postdoc, found it independently, Hiro Nakauchi, that in mice, as you get older, the blood-forming stem cells tend to give you myeloid biased output. When you're young, it's lymphoid or balanced, but when you're old, it's myeloid biased. And we went through to show, Wendy Pang and I, that it was not only true in humans, but all you had to do was look for that cell, the common lymphocyte progenitor, which is the first in the pathway between the hematopoietic stem cells and all lymphocytes. You just enumerate them as part of the whole bone marrow. And they have mainly disappeared. When I saw that, I thought, okay, way back when I was starting off in immunology and reading papers, the craze then was the clonal selection theory of immunology, that somehow you had to account for, you could be vaccinated against antigen A or have been infected with measles virus. You're protected for life because you have immune cells that have expanded. The clone that expands has a receptor for that antigen, but not for any other antigen. So clonal selection theory was a huge, huge thing. And so that led us to say, well, it must be that you need new lymphocytes to be produced for life in order to make it. And since we'd found this myeloid bias with aging in both mice and humans, and the last common progenitor between mice and humans was 90 million years ago, then it became clear the pattern of what this was going to tell us. And the first part of the pattern, the one that people hardly ever get, but I'm going to say it to you, is that it was okay for your lifespan 90 million years ago, 90,000 years ago, 900 years ago, if you met new pathogens where you lived when you were young, because you would make clones of memory T and B cells, and that's what protects you. And those memory T and B lymphocytes live as long as you do, whether they do it by self-renewal, but they do live as long as you do. But trains, planes, and cars bring you new pathogens. And so you just think back, the devastating effect of HIV and Zika and every flu epidemic, and of course, coronavirus. I even got together the immunologists and the microbiologists at the National Academy of Sciences, and I said, this is a problem for old people. They have to be able to rejuvenate their immune system or be vaccinated against it or something. And also, because of the homing receptors that Gene Butcher and Mike Galvin and I and Bernard Holtzman had discovered, some homing receptors on lymphocytes give rise to the plasma cells that occupy the oral and pharyngeal mucosa, and they're the only ones whose antibodies make it across the mucosa and protect against the viral infection. Integrin alpha-4 beta-7 is the homing receptor that takes them there, but it doesn't take them to the lymph nodes or to any other part of the body. So what do they vaccinate? The arm. They're L-selectin. They're great, markers, but they hardly go to the mucosa. So the reason I'm saying this is the brightest people on earth in both fields, and I would say I felt like I was talking to an audience that didn't appreciate or didn't remember. I'll just say that. So it's still a problem that people learn, they memorize what they think is important, they memorize it from important other people. And even if they're experimenters, sometimes they can't cross over in another field. So I'll piss everybody off eventually with this. But it also pushed me in my lab to ask, why do you have myeloid bias when you're old and balanced when you're young? And as we went back earlier and earlier and found markers on the cell surface of myeloid bias, not on the balanced ones and vice versa, in mice at least, at all ages, you still have in the very few, but some myeloid bias stem cells with the markers, and lots of balanced ones. When you're old, it switches. So that meant, at least to me, is they might be able to compete for common niches, and that if we could get rid of the old ones, we might rejuvenate the blood-forming and immune system. And that's where Jason Ross, Weill Cornell, MD, PhD grad joined my lab, and we went through that in mice and published it in Nature this year, that it works. And the markers we've seen in mice, at least some of them are in humans. I have a postdoc and a graduate student now taking back blood-forming stem cells in mice earlier and earlier to see when that split occurred. And this is like another story. So when I was up for tenure at Stanford, my department chief called me in one day and he said, the leading immunologist at Stanford will not support you for tenure. And so that made me wonder if maybe I shouldn't have done an internship and a residency and moved to Montana, like where I came from, and fly fish for life. But luckily, a couple people at external places, when they heard I might leave Stanford, offered me a job. And just like the function of a company is to make a profit. Well, the function of a university is not to look stupid by losing people who succeed elsewhere. But that meant I got a sabbatical. And I decided I wanted to follow up on Ray Owen's study of where blood formation comes from. Malcolm Moore and John Owen, then Malcolm Moore and Metcalf, saw that the yolk sac was the first place that had blood in it. And so it was reasonable to say, well, if they have blood, maybe they have blood-forming stem cells, and maybe the cells will leave the yolk sac and go further. I'll just say this as quick as I can. So I worked in Oxford in a lab that Jim Gowans gave me. I had worked with him also while I was in medical school, and the late Jim Gowans, and with Richard Gardner and Ginny Papiano, they taught me how to get out yolk sac blood islands, how to make needles. And so I transplanted pre-circulation, meaning the heart hasn't even started pumping yet, the blood vessels aren't joined. I took out the yolk sac blood islands from an A by B mouse, transplanted them into the same age of a B by C mouse. They share B, different in A and C, and showed when they grew up that a very significant part of their blood forming system had come from the original yolk sac donor. Then a whole bunch of very famous people, still the most popular, you read the textbooks, it'll say, said, well, you know, it isn't the yolk sac that does anything for the adult. The yolk sac is for embryos. And it's going to be the dorsal aorta or hemogenic endothelium or this or this or this or this. So part of my reason of wanting to find that switch between myeloid biased stem cells and other ones is to be able to go all the way back to the yolk sac blood island. And I'll say, so far as we've gone back and we can study by adult transplants, they look to be separate populations. And the populations we have in the yolk sac should have done what I actually had done in experiments in 1964. Oh, no, excuse me, 1975 was the 10-year time. So anyway, I remember talking to Jim Gowans about it. Later, he came to visit our lab and saw that I was still working on blood formation like I had done in 75 and so on. He says, old habits die hard. And he was right. So I guess the lesson from that is there's something way more fascinating now about how blood formation generates from mesodermal precursors. What is happening to turn on that population of cells? How diverse are they from the beginning? And of course, I'll stick with the yolk sac is the original source of blood formation for adult hematopoiesis, at least from the experiments we did in mice. Now, I promise you, when I try to publish lineage tracing experiments that will show the same, I'll be rejected because it doesn't fit with what the other people have shown. We can take a bet on it, you know, might even get the paper out in time that it would be with the podcast.
Well, we'll have to have you on maybe in the future, not too long from now, to have a follow-up. I mean, there's so many lessons there, Irv. I mean, that was just such an incredible journey. I mean, some of the things I took away, first and foremost, like you mentioned, sometimes you got to think outside of the textbook, right? Don't be bound by the dogma that's been presented in the textbook or has been reiterated by however many folks in the field, right? You have to be inventive, you have to be creative, you have to think outside the box, right? Also, it's just a testament to your determination. In spite of having that grant that was ranked 64 out of 65,
you still push forward with that, and we're able to convince folks that what you're working on is worthy of funding. I wish I had that kind of influence, but maybe one day. I think that also speaks to some of your gifts as a connector, right? You mentioned that you're the one who wrote the original Prop 71 for CERM. It's happened in large part thanks to your fearless efforts and your determination. You're able to bring together clinicians, basic scientists, funders, philanthropists to make this CIRM, California Institute of Regenerative Medicine dream a reality. And it's perhaps been the biggest influence in making California the best place in the world to do stem cell research and regenerative medicine. I mean, in addition to the billions and billions of dollars for funding basic translational clinical stem cell research and therapy, it's also provided a lot of opportunities for students and trainees, myself included, actually, ranging from high school students to postdocs to learn about, stem cell biology and perhaps become clinicians and scientists themselves. And since CIRM supported Stanford Stem Cell PhD program, I'm also one of those CIRM supported former trainees, right? And like we talked about, you had such a big role in making CIRM a reality in the early 2000s, but did you ever think it was going to be around for this long? Could you just reflect on CIRM for a little bit, what it's meant for stem cell biology and what it's meant for your work too, even though they've rejected a few of your grants?
Yeah, I of course, the timing of it was, I knew already the beginning responses of those metastatic breast cancer patients was remarkable. But I also knew that the big commercial outfit wasn't going to do it. So in talking to people eventually, I thought we need to be able to study blood-forming stem cells. And the way we got human blood-forming stem cells was be able to create the environment from human fetal tissues. That's an issue. And I had been head of a National Academy of Sciences panel for cloning and stem cells. So, of course, even then in 2000, 2001, there were people saying, we'll charge you just $500,000 and we'll clone you. You'll have a kid. And some people in Congress got up and said, we're not going to let those people, those mad scientists, clone mutant armies. As if they would be born, grown up already. So enough for trying to do that in the federal government. But we had good advice about what to do in the state, because California is a state that if you said we are going to retain a woman's right to decide to end her pregnancy for her health or whatever reason, and that we could find a way without interfering or paying for the woman or the doctor or anybody else to make money out of it, then you could do it. And that turns out to be, and I think that's an important point, it turns out to be the way you can do fetal tissue for research. But then, of course, it was embryonic stem cells as well, and clones of people. So it turned out that one of the legislators in California, state legislatures, when I said, but I had my own panel of eminent people, and I would say, draw an embryo. And they always drew a fetus. They didn't know the difference between an embryo without a formed organ and a fetus, which is on the billboards. And I said, well, what's the consequences of that? He says, you shouldn't worry. I won't give his name. He said, in California, 60% of the people are in favor of the right to choose. And so you would lose in California if you took away the right to choose. So it became pretty clear that I didn't even understand the politics at all of what this situation would be, but there were other people who had data-based politics that they could do. So two things then. One is as a way to be able to take discoveries all the way, if you had a mind to understand whether they could translate. Another thing was that you could do clinical trials in academic institutions, so you didn't have to sell it to a company too soon. And later I found, and it did work for a couple of our things, I'll use the terminology of the VCs, the pre-money valuation of your discovery goes way up the closer you get to a practical output. And it should be. So that you could have, as incubators, academics, so long as they understand that the academic enterprise was not doing this to make money. They had to separate it and license out to whoever would do it. And so that seemed to help, but there's still all the people who were brought up in another way, and even people who had taken care of our breast cancer patients, who had begged a couple of the patients not to go into our trial. When asked by another company that I started to try to do it on my own money, and knew those patients were out many years beyond their expected lifespan by what they were going to offer with palliative intent, more humanistic therapy, they told the board of directors of that nascent company that I started, it won't work. And they knew the people who had already gotten through it and lived beyond the time they should have. So we have, I hate to keep bringing up problems, but you got to be aware of those problems. It's not just science that will convince master clinicians about what you should do or not do next. So anyway, we've gotten through that. We're getting down the road. We'll probably do sickle cell after breast cancer. Hopefully the breast cancer will work just as well. And the big lesson, I hope, will be that NIH will look at this. So remember, in 2001, when we were writing up Prop 71, George W. Bush, right after one of our public meetings, where we had the people who said, give me $500,000, I'll clone you. Or the scientists, on the other hand, saying, you know, we've tried to clone mice, tried to clone even cows, and you know about Dolly, but you don't know that 99% of the clones die. And they don't die just when there's normally a miscarriage. They die anywhere during the pregnancy, and if they die late, they often take the mother also. So it was against the Nuremberg Accords of how you do experimental medicine with people. And so we were very clear on that, at least trying to establish that you could do those experiments and that in a place like California, you get it going. So what was the next thing that bothered me most is that when I was asked to go on a study section a couple of years after I got tenure, I remember going to the first study section, immunobiology it was called. And I wondered how they let me in that room with all those smart people. And then somehow, and for some reason, that drifted away from just trying to get experts. And I have a pretty good idea. People worry about the old boy network. They worry about favoritism and so on and so on. So CIRM was set up in the law that they should only have expert review, and they should go to the top experts in the field to get reviewers, no matter where their status was in terms of assistant, associate, and full professor. But bet on the people who know better how to make a discovery and then what you might do after. And like any bureaucracy, that drifted. And I think sometimes to myself that maybe we were 64th because of that side of my life instead of the other, but you never know. So the point I'm trying to get to is there's a whole bunch of things we did in that experiment, CERN, that the federal government could do and become more efficient and get the development of a discovery toward a therapy so that you lessen the risk and increase the benefit. And even at the late stage of clinical trials, you can turn them around. I'll just say that, if you know what's going on. But you can't turn it around if your only function is to say the function of a company is to make a profit. I can save us 50 to 100 million if I stop it now. And who will care if I don't make a therapy? So somebody's going to have to shepherd it, even if the federal government could do it. I mean, right now, one of the people I respect the most, Tony Fauci, is the target of a whole political party. So he carried us through those epidemics, including that one, COVID, that I believe our discovery of depleting the myeloid biocells will rejuvenate them to the point they can be vaccinated. 90% of the people who died during COVID-19 epidemic were old folks. It's obvious they couldn't make an adaptive immune response to the virus. So knowing that, you would want to think, how can we get that to the point that it becomes a practice so that it will have a chance of making it? So that's where I think CERN is going. I think telling the discoverers that it isn't over when they make the discovery, they at least have to put their minds to the idea of what could be translated, because they're the only ones on Earth who will have that potential insight at that time. Providing the monies that they can participate and help form a team that is a not-for-profit entity so that you can do those early phase preclinical and clinical trials. And by the way, even though I had started a ton of companies, was on the board of many companies, did help write what was going to be the clinical trials. When we did it on our own, Ravi Majeti and I were there with the right people we could bring in because of CERN. I learned so much of where things can go right and where things can go wrong in clinical trials. And we still don't teach that in Stanford Medical School, and I'll bet you Weill Cornell doesn't either.
Right, not.
Yeah, and it is the point that medicine and medical research and translation of medical research could get way better, way more efficient if you would allow things to go further before commercial entities have essentially the right to buy them. When a company shuts down something, they don't go back and say, well, we shut it down, especially if they bought a whole other company that's gone. You can't retrieve that. So the longer you can keep it going with smart people doing experiments and smart clinicians doing clinical trials, I think the better.
Yeah, I think, what you're describing here, I think it's a lesson that most scientists don't get, as you said. They don't teach it, right? Because it's goes under the radar, or you want to ignore the harsh reality of that fundamental conflict between a company and, a scientific endeavor where the intent really is genuinely in earnest to save lives. That's the end game for all of us. Every scientist I've ever talked to, they have one lifelong ambition. It's not to get rich, maybe some of them, not the ones I talk to. It's to have their science be used in even a single person. I'll tell you, that would be my crown. If a single person is helped by something that I've done or I've discovered, then that would fulfill my lifelong ambition. But of course, the investors don't really care about my lifelong ambition. And the reality is that they are the ones driving that translation because they have the resources, right? But I mean, I would say even in earnest with CERM, which is, I think, a tremendous model moving forward, as you described, for kind of addressing that. Even so, best intentions, all that being there, sometimes it doesn't work out, right? And sometimes I think it's a matter of being a little bit ahead of your time in terms of concept versus how you can actually translate the concept or discovery into practice. And that was particularly the case in any nascent field. That's the case, right? A lot of challenges that are unrecognized and yet to be overcome. But in the stem cell field in particular, because the promise was so great and the need was so great and unmet, that maybe there were, I wouldn't say over promising, but maybe over hoping, if that's a thing. And I think a lot of people acted on that with best intentions. Some of those efforts didn't work out, but here we are, a quarter century later after the first generation of pluripotent stem cells, and it feels like industry is actually, on the way to realizing a lot of whatever their motivation, financial or saving lives, let's say both, they're on the way to realizing a lot of the translational promises that the public has in particular been waiting for. And it's been especially visible in California because of CERN, because there was the public little proposition, it was on the ballot. So as a seer, let's call you a sage, someone who's seen it all, but also is constantly looking forward and trying to bring these concepts back or forward into fruition. I'll ask you this, and I'm putting you on the spot here, so forgive me, but what pluripotent stem cell based, and I emphasize there, pluripotent stem cell based therapies, do you think are going to be the first to practically and broadly be used? And on the flip side of that, what do you think is going to take the longest to get into human use?
Sure. And I'll tell you something that will take so long it'll never happen.
Mm.
When we were doing all that, and as you said, there was a lot of public exposure. I know that I was very careful when I was in public audiences because I said, here's the time you have an approved drug. You got to have a phase 3 trial. Before that, you got to be successful in a phase 2. Before that, a phase 1. Before that, preclinical proof of principle. Before that, a discovery. That's 20 years. So it ain't going to happen for you. It's going to happen for your kids or your grandkids. But still, people listen to what they want to. And I'll just say, a whole bunch of people commercialized immediately. We have stem cells that can turn into anything. And what they had was cultures from bone marrow of stromal cells. So mesenchymal stem cells became a commercial product before anybody ever isolated a mesenchymal stem cell. So now, what's most likely to go forward? Well, I'm pretty sure that if we can solve that one step between mesoderm, yolk sac blood island, and transplantable hematopoietic stem cells, we're just, one step away from it now, then I think that will be the source of blood-forming stem cells. We already, and many others have, we isolated human fetal neural stem cells, but now many people, really top-notch scientists, can make forebrain plus midbrain precursors and hindbrain separate precursors. What I didn't tell you is that when we did the human fetal brain, whether it was in Dan Liu's paper last year or Nobuko Uchida's from 24 years ago, we were lucky. We put them into the brains of newborn immune deficient mice. We already knew about the lateral ventricle having right next to it a subventricular zone, which is a neurogenic zone for what's called the rostral migratory stream in mice. And so we put the cells in the lateral ventricle, and they seeded not only that subventricular zone to make inner and outer radioglia, they call it nowadays, but they seeded the dentate gyrus of the hippocampus, the one that Rusty Gage's lab has championed to answer one of the biggest questions, the one I marked on, and that is, how does the brain make the mind? You think about it, that we're having this conversation, you understand immediately what I say, I understand what you say, and in a microsecond I can, and you can, have the conversation still going, but we have no idea how that works. We're listening to the auditory part, just think of the other sensations. But the answer is in the brain. And spectacular advances in optogenetics for a long time in circuitry have laid the pattern of what it is when it's already developed. But they don't have any idea how brain stem cells can give rise to progeny committed to an excitatory neuron, an inhibitory neuron, an oligodendrocyte, or an astrocyte, and how those cells migrate absolutely accurately to create parts of the brain. So I would say, if there's one problem I wish I could solve, that's it. How the brain makes the mind. Because you look at the tragedies of people who get schizophrenia, manic depressive disorder, opioid addiction, any of a number of things that they can't stop. They can't do anything about. You know, the teenager who starts hearing voices, who later become persona in their head, and who never can be turned off, whereas the daily world they see, you just walk out of the room, you turn off all those others. You can't turn off the voices in the brain. So I got some hints about what it might be. Nature asked me in 2004 to write, from what I knew about what we had discovered, where could it go? You know, be expansive. So I picked schizophrenia and voices in the brain, and even a couple of examples. I won't go too far on this, but I said, you know, those voices are real. And in London, they showed in 1995 with PET scan that a real part of the forebrain, or the prefrontal cortex, flashes when a voice goes on. And if that same voice goes off, that flash goes away, but another one can happen in another part. So there's going to be a physical basis. And I know I'm going too far, but what, after a year and a half of them asking me to write this, they rejected it, and they said, you know, we talked to some neuropathologists. And if there was, I claimed that there could happen easily that a glutamatergic neuron mismigrates and is no longer inhibited by GABAergic neurons in the adult brain. And so this editor said, the neuropathologist told us they would have seen it. I said, seen what? Well, you think that there's a cluster of cells that might be in the clear space of the dentate gyrus, right? Not in the granular zones, okay? I said, don't tell me that crap. Way back when I had worked with Gowans on thymus cell migration while I was a medical student, I saw where they went, and I saw the lymph nodes and spleen, the Peyer's patches, collected them in one place, but there was another absent place. And I wanted to know what it was. I had to modify immunofluorescence of tissue sections so that I could see membrane antigens. And we were just barely getting to know there were different T and B cells. The separation hadn't occurred at the time we did that. So we developed that. So I told this editor, we developed that. And I was across the hall from the most famous lymphoma pathologist in the world. And I showed him those structures. And he says, "What's that?" They didn't know what a germinal center is, other than from a lecture from Bob Good, which was wrong at the time. They all, of course, only had memorized their way. I keep saying the same thing. It takes somebody who has a need to make a discovery to alter even a technique to make the discovery. And now I'm being told by neuropathologists that an idea of how the brain could make the mind couldn't possibly be.
No, I think just an unlimited number of questions stemming from that phrase, like how the brain becomes the mind. I think, you and many, many folks around the country and around the world are trying to tackle that. Because I think it is the biggest unanswered question in modern science. And it goes beyond just the nitty-gritty of the biology. It becomes a philosophical question as well. And I think we have to tackle both of those at the same time. So, Irv, this is going to be an amazing conversation for our trainees and our students to listen to. We're going to have you back on the show in the very near future, since your research takes many different avenues, and we could talk here forever, honestly. But at the tail end of all of our conversations, we like to ask our guests a couple of peripheral questions adjacent to the science, and you actually answered one of them. If the first one is, if you could answer any single scientific question regardless of your expertise or chosen field, what would it be? And your answer was how the brain makes the mind. So certainly a great answer. And the second one that we'll ask you is, what is the biggest misconception about science that you would like to resolve?
I actually answered that. And that is, how would you advise your 6, 8, 10, 15-year-old to become a scientist? And even the scientists say, get straight A's. When the answer isn't that. The answer is, get into a laboratory, figure out what experiments really are, and tell the people who run the lab, don't micromanage the people. Let them, once you've got them started, let them think about what might be an important phenomenon to understand, and then what kind of experiment they would set up. And that's a much more productive path, I think. But you go into even Stanford Medical School, I'm sure it's the same where you guys are. They'll say, well, the most important thing is you've got to mentor people. And I would say, no, keep your hands off people. Don't try to tell them what to do, because you'll rob them of the chance of figuring out how to ask and answer questions.
Yeah, I mean, look, this for me is such an inspiration for what's left of my career, because it's a validation of some of the great doubts I had about my approach, my capacity, my ability to succeed amongst, peers who are truly the smartest people in the world. And talking to you, which is not to say you're not amongst them, because clearly you are, and you at the output to prove it. But, I know your story, and I don't think you're someone who was a classic, scholastic high achiever. I don't think that it was really in you to seek those accolades and plaudits. I think that you were driven by your own curiosity, as I think you've shared over many interviews over the year, and your own, dogged and deliberate, maybe sometimes stubborn resistance to what people trying to get their hands on you, as you put it. So it's a real validation, I think, for you, and inspiration to all the young people behind us as trainees, but even at any level of science, that really you can do great things just by sticking at it, keeping your eyes open and thinking differently. And I think that's the most important and critical key, is that it's important to take what you can from your mentors, but you got to find your own way, and you got to have confidence in your own way of thinking. And that confidence shouldn't come from straight A's. It's not a prerequisite. It's just a reflection of your own passion and interest, and that can take many forms and manifest in different ways. So, Irv, once again, you've inspired me. I'm sure you've inspired a lot of our listeners, and Arun and I are really grateful that you take the time. The best thing, someone as busy as you with 7 or 8 companies and a whole scientific family to support and cultivate, that you take the time to talk to us and to everyone. I think it's the greatest thing about you. So thanks so much, Irv.
But as you know, the best part of running a lab is these conversations.
Oh man, absolutely. That's everything.
Yeah. Thank you.
Thank you. All right, you guys, that brings us to the end of our show. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at StemCellPodcast or by email at info@stemcellpodcast.com, where you can provide feedback or suggest guests. Thank you guys so much for listening. Until the next time.